So that's the lesson about that. And then we'll talk, like I said, we'll talk more about the actions of antibiotics and different types of antibiotics. We do all that stuff in chapter 20. And then, lysozyme. Lysozyme is one of my favorite things ever. And believe it or not, one of the most just amazing coincidences of all time, Dr. Fleming is also the person who discovered lysozyme. So the guy who discovered penicillin also discovered lysozyme. Amazing, right? Super amazing. So lysozyme, whoops, I kicked my table and knocked things around here. Lysozyme is an enzyme that our body produces naturally that can also damage the bacterial cell wall. So lysozyme is, it's an enzyme, but it almost works very similar to how antibiotics work, which is great. You should have learned about lysozyme in A&P1. Definitely you would have learned about in A&P2, but lysozyme is found in your tears, in your saliva, and in breast milk. So it's a very strong, very potent enzyme. And again, it makes sense, especially like your tears, your eyes are constantly exposed. As long as you're awake, as long as your eyes are up, bacteria can be getting in there. That's why you need that powerful lysozyme to keep your eyes, from getting an infection. Okay, so this is not the most exciting topic in the world. It wasn't the most exciting when you talked about an A&P1, unless you're really, really into it. But remember, the bacterial plasma membrane is nearly identical to what you would find in a eukaryotic cell. So you should have learned about the plasma membrane in A&P1. You've got your phospholipid bilayer. You've got your hydrophilic head, your hydrophobic tails, all that stuff. It's exactly the same in bacteria. So the components or the structure of the bacterial cell membrane is almost exactly the same as it is in animal cells. The one big difference is that bacteria do not have cholesterol. Eukaryotic cell membranes have cholesterol, which acts as a fluidity buffer. So it keeps the membrane more fluid. Bacteria don't have that. So that's the one big difference. Okay. So there you go. This picture should look very familiar to you, unless maybe if it's been a couple years since you took A&P1, you may have forgotten. But you've got your phospholipid bilayer, you've got your hydrophilic head, your hydrophobic tails, you've got your peripheral protein, your integral proteins. Everything's the same, right? So the plasma membrane of a prokaryotic cell, plasma membrane of a eukaryotic cell, just about exactly the same. Oh, who remembers passive transport? Scary, right? Because nobody likes this stuff in A&P1. So passive transport, it's the same thing as diffusion. It is the type of cell transport that requires no energy. So molecules or materials will just move from a higher concentration to a lower concentration. So wherever there's more of it, they want to get away from that. They want to be spread out. They want to have like their own little personal space, right? So passive transport is the same thing as diffusion. A lot of times we'll use the term simple diffusion when we're talking about things like oxygen and carbon dioxide. Facilitated diffusion is when you need a membrane protein to help. So facilitated means like you need a helper, you need a facilitator. So this would be like if you're trying to move things like glucose in and out of the cell, iron, what's the other one I use example all the time? Calcium, that kind of stuff. And then of course, water is so special, it gets its own term, and that term is osmosis. So we say, you could say the diffusion of water, that's fine, but osmosis is the specific term that we use when we talk about the diffusion of water. So if there's less water in the cell, water is going to move in. And if there's less water outside the cell, water is going to go out. And that's where this stuff comes in. So again, definitely a topic you should have covered in A&P1. We're going to do a little bit of a reminder because just like eukaryotic cells, prokaryotic cells are affected by their environment. And similar to us, animal cells or human cells, however you want to look at it, prokaryotic cells want to be in an isotonic solution. Isotonic solution means that the amount of water and the amount of solute inside the cell is equal to the amounts outside the cell. ISO means equal. So an isotonic solution, everything's equal, there's no movement of water, and the cell is happy. So if it becomes a hypotonic solution, is when there's less water in the cell. So water is going to leave the outer environment. It's going to go into the cell, and it's going to cause the cell to swell up. Now, bacterial cells, one of the main jobs of their cell wall is to prevent them from bursting. We call that lysis. So if the hypotonic solution isn't so bad, if it's not so strong, then the bacterial cell might swell up a little bit, but it won't explode the way our cells could explode because we don't have a cell wall. And then hypertonic solution, that's the opposite of hypotonic. So water is going to leave the cell, go outside of the cell, and the cell will shrink. So hopefully you remember all that stuff from A&P1. It's all the same. The one big difference, of course, is that prokaryotic cells have a cell wall, and that cell wall does give them some added protection in a hypotonic environment. So that's the one big difference here, because of course we're talking about bacteria. We're not talking about eukaryotic cells, which is what you would have talked about in A&P. And then active transport requires energy. It requires ATP. Materials are forced to move against their concentration gradient, which means from a lower concentration to a higher concentration. Again, all the same that you should have learned in A&P. And then I just put a reminder here. I know that when in my A&P1 classes, when I talk about active transport, I just always say, just think of the sodium-potassium pump. The sodium-potassium pump is the only example of active transport that we really, cover. Like if you were going to take a higher level cell biology class or cell physiology or some kind of thing, then you would talk about more of this stuff. But really, let's keep it simple. Sodium potassium pump, active transport, same thing, and just move on from there. All right, so you should know what cytoplasm is. Cytoplasm is all the materials inside the cell. So water, protein, ribosomes, all that stuff. And then, of course, we already talked about the prokaryotic cells do not have a nucleus. They have a nucleoid. The nucleoid is the region of DNA. The bacterial DNA is usually just a single long strand of DNA that wraps up into a circular shape. So instead of having what our chromosomes look like, right, which they have like the little lobes, the little, I don't know, they almost look like a little tiny propeller. Okay. Bacteria is just a circular shape. It's just literally like, looks like a little wheel, little circle. And then I didn't talk a lot about the plasmid at all. We're not going to get into plasmids too much, but they're kind of cool though. A plasmid, it's like a little separate circle of DNA. It's not, it's like set off from the nucleoid. So the nucleoid's like this, big region of DNA. And then off to the side, we have the plasmid, and the plasmid's like a little tiny circle, like a little tiny ribbon of DNA. And what it does is it can potentially give the bacteria some new features, some new abilities. But the other thing that can happen is bacteria can actually exchange plasmids with each other. And that can lead to, some adaptation, some bacterial evolution. And the other thing is it's used extensively in genetic research, because what we can do is we could actually remove a bacteria's plasmid and we can inject it with a new plasmid. So we're basically like injecting it with the DNA that we want to inject into it. And you can do a whole mess of things when you have that, that technology available. All right, so then inclusions. Inclusions is kind of a new thing, or at least something you definitely didn't learn about in A&P. So inclusions are various different types of reserves or deposits found within the bacterial cell. There's a whole bunch, right? So we're going to go through this list. Some of these will be on the test, some of these won't be. Excuse me, sorry. The first one is called velutin. Velutin are metachromatic granules, which means they have like a color or like a sheen to them. They are reserve deposits of inorganic phosphate. So it's basically, velutins are like storage of inorganic phosphate. Does anyone know what inorganic phosphate is used for? What do you think? What do you think? Inorganic phosphate. Inorganic phosphate is used in the production of, does phosphate ring a bell for anybody? What if I said adenosine triphosphate? So inorganic phosphate is used to make adenosine triphosphate. A T P, right? ATP. So ATP stands for adenosine triphosphate. We know what ATP is. ATP is energy, right? So bacterial cells have these special little storage compartments inside of them that store phosphate that is used to make ATP. And then the other thing is this type of inclusion is actually used to identify the bacteria that causes a disease called diphtheria. You guys have probably never heard of diphtheria before because it is a vaccine that we've been using for decades. It is an extremely rare disease now in the United States because of that vaccine. But 50 years ago, it was very common. Polysaccharide granules, that's not a surprise, of course. So we know that glucose is very, very important to the bacterial cell. It's important to our cells as well, right? So glucose is used for the same thing across the board, right? We take the glucose, we rip it apart, and we turn it into energy. Now, our cells, it's a very long, very complex process because we need to get every last little tiny drop of ATP out of every single glucose molecule. We are very complex organisms. We require lots of energy, right? We have lots of cells, and those cells have lots of mitochondria. Mitochondria make the ATP for us, okay? In AMP2, sorry, I keep saying ATP and AMP, and it's probably hard to keep track of. So if you haven't taken Anatomy and Physiology 2 yet, there's a whole chapter on that. It's called Muscle Metabolism. And you basically learn all of that biochemistry that's involved with making the ATP out of the glucose. Now, we don't need to go into all that crazy detail, all right? But just, you know, if you haven't had AMP2 yet, that's going to be something that you talk about in that, you know, in that chapter or in that unit. So for bacteria require much, much less glucose because they don't need as much ATP. They're not nearly as complex. But just like us, bacteria like to eat, and they like to eat things that are carbohydrate. So that's why things like, you know, things rich in sugar, things rich in starch, you know, apples, potatoes, cereal, bread, pasta. Like these are things that kind of can go bad pretty quick because the more the bacteria love to eat it, the faster, you know, it's going to spoil. It's going to go bad. So glycogen is how animal cells store glucose, and starch is how plant cells store glucose. Well, bacteria can store either one in these granules. So whether the bacteria is eating something that's an animal-based or a plant-based food, doesn't really matter to bacteria. If the bacteria is going to ingest starch, if it's going to ingest glycogen, and if it ingests more than it needs right then and there, it can store the two of them. So it's like food storage. It's like a pantry almost for the bacterial cell. Lipid stores, which not all bacteria, of course, are going to have lipid stores, but obviously that's another type of food item that the bacteria might want to store. Sulfur granules, so lots of proteins are rich in sulfur. And then, believe it or not, some bacteria can actually use sulfur in its metabolism. We'll talk about this in a couple chapters. You guys probably aren't going to like that chapter, but that's okay. But we'll talk a little bit about this because this is something that we really can't do. But bacteria can use sulfur in some of its metabolic processes, and that's why you get that stinky smell. So sulfur is like the rotten egg smell. If you've ever had the horrible opportunity to smell rotten eggs, it's really bad. If you don't know what I mean by rotten eggs, it's like really, really bad fart smell, basically. So that's why, like, sometimes, like, you know, things will stink, especially things that are rotting or decaying, because the bacteria is eating away at it and it's releasing gases. And one of the major gases it releases is sulfur gas, and it stinks. It stinks terribly. So that's part of the reason why when things are rotting, they stink. It's because the bacteria is giving off all these weird gases. Carboxysomes are a type of inclusion in photosynthetic bacteria. If you remember way back when, I told you the term cyanobacteria. So that's the photosynthetic bacteria are cyanobacteria, cyano bacteria. And they're called that because cyan, it's like that bluish green color. It's almost like the same as teal. That's how they get their name, because it's bacteria that have a bluish green color because they're photosynthetic. Bacteria that live in water, they have gas vacuoles that they use to kind of move through the water so that they can kind of float and sink and float and sink and float and sink. They use gas vacuoles for that. And then magnetosomes are storages of iron oxide that some gram-negative bacteria have. It's used for certain types of motility or movement. And then it could also provide some protection from peroxide accumulation. So again, back at AMP1, your teacher may have told you about something called a peroxisome. A peroxisome is an organelle that helps to detoxify peroxide in our cells, because our cells don't like peroxide, bacteria cells don't like peroxide either. But this is one way that some gram-negative bacteria are able to combat the accumulation of peroxide. So, I would just make flashcards of the different types of inclusions. I know for sure that not all of them are on the test, but some of them will be. I'm not going to tell you which ones, obviously. So you just got to study those. But I would say mostly just treat them as vocabulary words. They don't have to be any, like I know we spent a little bit of time on the first couple. It's not like it's going to be some big, huge, long explanation. It'll be a multiple choice question, and you just got to pick out the right one. So don't spend a lot of time on these, but certainly you should know. Just treat them as vocabulary words. They are important when you're talking about, bacterial cells. Inclusions are one of the main kind of topics. All right, so now we're going to talk about endospores. Endospores are a very important topic for us because they are related to some very dangerous diseases. So endospores are a specialized type of cell that it's mostly gram-positive bacteria. It's not always gram-positive, but again, we're going to try to keep things simple. We're not going to overcomplicate stuff, okay? So we're just going to say endospores are produced by some gram-positive bacteria, okay? If you remember the taxonomy, kingdom, phylum, class, order, family, genus, species, these are two genuses. We don't say genuses, we say genera, but whatever. So Bacillus is one genus of bacteria. One bacteria name, you can get to know this one, Bacillus anthracis. So Bacillus anthracis is the scientific name for the bacteria that causes anthrax. And of course, anthrax was the very first, disease discovered that proved the germ theory. So that's what Robert Koch was working with. He was working with anthrax, and he proved that anthrax is a disease caused by a very specific type of bacteria. That type ended up being named Bacillus anthracis. Bacillus, because it's a bacilli bacteria, that's what shape it is. Anthracis, because it causes anthrax. Then we have Clostridium or Clostridial bacteria. So Clostridium includes two significant diseases, tetanus and botulism. Another one, which we're not going to spend as much time on, but it's also a clostridial disease, is gangrene. So anthrax, tetanus, botulism, and gangrene. Those four diseases can be linked to endospores. Okay? So bacillus is what gives us anthrax. Clostridium is what gives us tetanus, botulism, gangrene. All four of those diseases can be caused by endospores. And you're like, okay, what are endospores? Okay, so what happens is the bacteria is living its life. And for whatever reason, conditions become bad. So there's no more water. There's no more food. It could be very hot, very cold, very dry. It's just life is no longer good for the bacteria, okay? The bacteria just happens to be living somewhere where all the water has dried up. There's no more food. And the bacteria is like, well, darn it, I guess I'm going to die. Except if it's a type of bacteria that can make endospores. What the bacteria does is the bacteria literally inactivates itself and turns itself into like this little shell. So it's almost like a little egg. But it basically, the bacteria forms this thick shell around it, and it just sits there and waits and waits and waits. until conditions become good again. Endospores are the most resistant biological structures known to man. It's very significant. It's very crucial to understand that. The most biologically resistant structure that we know of, endospores. Most types of endospores, you could boil them. They won't die. You can dry them out. They won't die. You can spray them with Lysol. You can drown them in bleach. They won't die. They are very, very resistant. You can irradiate them with gamma rays and they won't die. They are extremely resistant. And in fact, there's really only two things that we know of that can kill endospores. Fire, so you could set them on fire. If you set them on fire, they will die. Or chlorine gas. And that is how powerful chlorine gas is. It really gives you a respect for chlorine. So, most of you are too young to remember, in the weeks and months after 9/11, there was a bunch of anthrax that was sent through the postal system by terrorists. And, these letters and packages were going out to, senators and governors and all that. And again, anthrax is one major endospore former. So what they had to do is they, of course, had to evacuate the post offices, right? The only way to kill these things was either to burn the post office down to the ground or use chlorine gas. And if you can watch, if you watch the news back then, they had these people in these biohazard suits, and there was this they would have like this kind of tube, and this white smoke was coming out of the tube, and they were just going everywhere spraying this white smoke everywhere. That white smoke was chlorine gas. They had to be in that biohazard suit because if they breathed it in, they would die too, because chlorine gas kills everything. Humans, plants, animals, fungus, mold, bacteria, viruses, everything gets killed by chlorine gas. The reason they had to use it was because it's the only thing that we know of that works on anthrax endospores. It was either that or burn everything to the ground, because fire works too. Fire works on everything. You set something on fire, it's going to die. The scary thing is endospores can potentially be dormant forever. There have been endospores that have been excavated from ancient Egyptian tombs. You're talking 5,000 or 6,000 years old. These endospores are still viable. You put them in a little petri dish and they come back to life, right? Most of us, we know how we get tetanus, right? You get tetanus from cutting yourself on some sort of dirty, rusty, piece of metal, or, something that's been sitting around for years and years and years, and it's been collecting dust and rusting over, and it's like it's crusted over with the, with time. That's when you really have to worry about tetanus, because tetanus is another endospore disease. So, you might, you're in a junkyard somewhere and there's a piece of metal or a board or something that's been sitting there for years and years. It could have tetanus endospores on it. And when you cut yourself on it, the endospore gets inside of your cut and it says, woohoo, I'm free, right? Because your blood has water, and glucose and protein, and it's warm, and the bacteria says, oh, this is a place I want to live. And that's how people get tetanus. Tetanus is nearly 100% fatal. That's why it's so important to get your tetanus shot, right? So the tetanus vaccine is another very important vaccine that everybody should get. You are supposed to update it every 10 years. So every 10 years, you're supposed to get a tetanus booster. Most people don't do that, right? But what happens is, if you're, anytime that you go and get stitches, unless, you know, it's documented that your tetanus shot is updated, they're going to give you another tetanus shot, right? It's still an optional thing, but anyone who turns down a tetanus shot, not good, okay? So if you're in an accident and you have cuts and you have wounds that have to be stitched up, you have to get stitches. Even if it's, you just kind of fall down and hit your head and you get one of these kind of gashes, right? But the bleeding is not stopping and you have to get stitches, and you got to go to the hospital, or you got to go to like medical aid unit, you got to go somewhere and get stitches. They are going to give you a tetanus shot, or at least they're going to say you need a tetanus shot. Always a good idea to get that done. Even if you're like, well, I'm pretty sure I got that done like 4 or 5 years ago, just get another one. I'm telling you, just get another one. Tetanus is a disease you do not mess with. Tetanus is deadlier than anthrax. So get your tetanus boosters. All right. So there you go. Some very serious, very scary diseases on this list. The worst is tetanus. And we will talk about all of these this semester. We will talk about tetanus and botulism in even greater degree. We will definitely talk about those for sure. We'll talk about all 4 of these, but tetanus and botulism we'll talk about extra. And like I said, very, very difficult. You either have to set them on fire or kill them with chlorine gas. This is crazy if you think about it. Think about you have, I don't know, you have a knife, or you have a coin. I don't know. You have something and it has endospores on it, right? And you're like, it's okay, I'll just clean it with bleach, or I'll put it in boiling water. It doesn't work. That's crazy if you think about it. Putting something in boiling water and it won't die. Bombarding it with, ultraviolet radiation and it doesn't die. Like, that's pretty crazy. But that's what endospores are. So here's the lovely picture in the book. Now, you're not going to have to label or identify or describe any of this. I just like to show it to you so you can understand how these endospores form. This is on page 93 of the book. So basically, up at the very, very top where it says number 1, you've got the cell, and the cell is like, oh no, it's dry and there's no food, there's no water, and I'm living in a terrible place right now. If it happens to be a type of bacteria that can do this, because remember, not all bacteria can do this. This is a special thing that only some bacteria can do. What they're going to do is they're going to replicate their DNA. They're going to double their DNA, just like how you learned about in AMP1. And that, well, no, because it's a little bit different, but whatever. So the DNA is duplicated, and they basically just kind of surround the DNA in a membrane, and then they just keep doing that. And then they form a peptidoglycan layer around the, between the membranes, and then they form this spore coat around the peptidoglycan. So it always makes me think of like an egg. It's like the cell is like kind of making like this little egg kind of thing. But it's called an endospore, and it's just, and it's not the same thing as a spore. A spore is like a mold spore. And that's how like a lot of different types of fungus reproduce. So an endospore is not the same thing as a spore. Okay, so just don't get those mixed up. We'll talk about spores a little tiny bit when we talk about mold. Okay, so that's the endospore of anthrax. And it's just this, it's literally just this little tiny thing that it just sits there. And it can just sit there forever until finally conditions improve. So now all of a sudden there's water and there's food and whatever, and the bacteria is free again. But of course, this has some major disease ramifications. So, you know. All right, everybody okay so far? Questions? We good? We awake? All right, let's keep on going here. Now we get to the eukaryotic cell. And this is where I said I am going to go quite a bit faster, because a lot of this stuff you should remember from AMP1. If you don't remember it from AMP1, you're just going to have to relearn it again. So who are we talking about? We are talking about fungus, algae, protozoa, or parasites, right? They all have eukaryotic cells. And of course, so do we. So you have to know about the eukaryotic cells because, you know, your patients are going to be humans. So you have to know about the human cell and how it works and everything it does. And then you have to know about the cells of the enemy, which is mostly bacteria. You know, viruses, of course, are a whole nother kind of crazy thing. I was just saying last night, I was talking to somebody and I was just like, viruses are, and I said some words that I can't say in class, right? But, you know, viruses are jerks. They just, it's ridiculous. So we're going to talk about viruses next class. You know, that's chapter 13 topic. But I'm telling, at least bacteria, they have a job. You know, bacteria, they're living things. They're made up of cells, right? They, you know, they eat, they breathe, right? They need oxygen, they need glucose, they need ATP. You know, some of them have really important jobs in nature. You know, some of them, they're involved in the carbon cycle, they're involved in the nitrogen cycle. They may, you know, provide nutrition for other organisms. You know, say what you will about bacteria, but only about 1% of all the bacteria on Earth can make us sick. And they have legitimate jobs. They have legitimate lives to live, right? Viruses are nasty, mean little things. And their only reason to exist is to kill cells. Like, could you imagine, like, your only reason to exist is to kill things? Like, that's pretty terrible. That's a pretty horrible way to be. And that's what viruses are. So we'll come back to that. But I just, again, you know, say what you will about bacteria, but they are at least like kind of our fellow living things. The viruses are just mean. They exist to kill things. They're pretty awful. All right. So eukaryotic cells, much larger, much more complex than prokaryotic cells, probably evolved from different types of prokaryotic cells, you know, just hundreds and hundreds of millions of years ago. And of course, plants and animals are also eukaryotic organisms. Eukaryotic cells have a nucleus, that's where the DNA is found, and they have organelles. So we know that the nucleus is the control center of the eukaryotic cell. It contains the DNA. DNA associates with proteins inside the nucleus to form a material called chromatin. The nucleus is enclosed by its own special membrane. You can call it the nuclear envelope or the nuclear membrane. Doesn't really matter either way. It's the same thing. The nuclear membrane has holes in it that are called nuclear pores. Those holes are there so that the mRNA has a way to leave the nucleus to go find a ribosome. So hopefully you remember from AMP1, you've got your double-stranded DNA They have bases that pair together. So A goes with T, C goes with G, right? And then through a process called transcription, the DNA makes the mRNA. The mRNA is single-stranded. It leaves the nucleus through the nuclear pore. It goes and finds a ribosome. And then in a process called translation, proteins are made in that ribosome using the mRNA. That's how that whole thing goes. So the rough endoplasmic reticulum, or the rough ER, that's where the ribosomes are found, and that's where proteins are synthesized. The smooth ER is smooth because it doesn't have ribosomes, and it synthesizes other non-protein items. The Golgi apparatus, I always like to call the Golgi like the warehouse. So it packages, sorts, modifies proteins that are synthesized by the rough ER. So the rough ER has the ribosomes. The ribosomes are like the workers that make the protein, and then they send the proteins to the Golgi. The Golgi figures out what to do with them next. Lysosomes are probably the organelle we care about the most in our class, because lysosomes are what the cell uses to kill bacteria. So lysosomes are sacs of digestive enzymes which help to protect the cell by digesting the bacteria that tries to get into the cell. So we're going to talk a lot about this process. We're going to talk about phagocytosis and how white blood cells in particular have this ability. Of course, we all know the mitochondria is the powerhouse of the cell. It makes energy, or ATP, using glucose and oxygen. Mitochondria have two membranes, an inner and outer membrane. And then you may have learned about in A&P how the mitochondria also has a role in apoptosis, which is programmed cell death. And apoptosis is a very important thing that is being studied, especially in cancer cells, because if we can trick cancer cells into killing themselves, that would be great. And that's a big area of research right now, trying to get cancer cells to basically self-destruct. And the chloroplast you did not learn about in A&P, or at least you definitely shouldn't have. The chloroplast is what plants and algae have in order to do photosynthesis. And plants most likely evolved from a special type of green algae. So the endosymbiotic theory states that eukaryotic cells evolved from prokaryotic cells. It is believed that smaller prokaryotic cells entered larger prokaryotic cells and became part of that cell. So they essentially established a symbiotic relationship. Those smaller prokaryotic cells basically evolved into things like mitochondria and chloroplasts, and they became integral parts of the larger cell's metabolism. And then if you want a little bit more explanation of the endosymbiotic theory, you can read about it on page 102. But as long as you just know the definition, the endosymbiotic theory states that eukaryotic cells evolved from prokaryotic cells. That's basically the definition. But if you're interested and you want to read more about it, it's in your book at the very end of the chapter 4. And speaking of, that is the end of chapter 4. So we did end up getting through it, and we have a little bit of extra time, which is nice. So remember